or the one on that?
Good morning, everyone. Good morning. Thank you very much for joining us today. The event today is in honor of the life and the scientific contributions of Professor Zev Pancer. And I would like to start off by asking you to join me in a moment of silence as we think about Zev. I serve as the Director of the Institute of Marine and Environmental Technology. We have been joined today by many distinguished people, but the first person I want to recognize is Zev's aunt, Gladys Siegel, who is with us representing Zev's family. Zev was very close with Gladys and spoke of her often. I have been fortunate enough to meet Gladys previously when she joined us for one of the fireworks celebrations here at IMET, viewing the fireworks, and we're delighted you could be here. At one point we thought that Zev's mother may be able to join us, but she was unable to travel from Tel Aviv, so Gladys is representing the family, and of course we're all thinking of Zev's mother today as well. I spoke with the president of the University of Maryland, Baltimore yesterday evening, and his thoughts are with us today as well. He is delighted that we are holding this event. This event is a scientific celebration, and we're going to have two outstanding seminars. This was the most appropriate way we could think of to honor Zev. So Professor Perman's thoughts are with us as well, and we are fortunate to have UMB very well represented here today. Bruce Jarrell will be here a little bit later. Bruce Jarrell is the Senior Vice President of Academic Affairs and the Chief Academic and Research Officer of the University of Maryland, Baltimore. Representing the School of Medicine here today is Professor James Caper. Jim is the Senior Associate Dean for Academic Affairs, and he also serves as the Professor and Chair of Microbiology and Immunology at UMB. We have also Professor Eckert with us. Rich Eckert is Professor and Chair at UMB. He's the John F.B. Weaver Distinguished Professor and Chair of the Department of Biochemistry and Molecular Biology, and that is the department with which Zev's academic appointment was. And I also thank Rich for all of his help and support over the last few weeks, and this event today is co-sponsored by the Department of Biochemistry and Molecular Biology at the School of Medicine. I'm very pleased as well to have Professor Lee Mao here. Lee is the Professor and Chairman of the Department of Oncology and Diagnostic Sciences. He's the Associate Dean of Research at the University of Maryland, Baltimore School of Dentistry. And Professor Mao was a close collaborator with Zev. He served as a co-investigator on Zev's NIH grant. So it's great that he could be with us as well today. Some of the people with us have traveled a long distance to be here, and I want to mention in particular Satoshi Sasumi, who was a postdoc with Zev in the past, and he is now in the fisheries laboratory at the Graduate School of Agricultural and Life Sciences at the University of Tokyo. So he traveled here from Japan to join us today, and I know some of the other guests have also traveled a long way to be here today. So I mentioned Satoshi, who worked very closely with Zev as his postdoc. We also have with us Xia Hong, who was the postdoc in Zev's lab over the last few years, who really had by far the closest working relationship with Zev and was absolutely indispensable in moving his research forward. It's great that she could be here. She will also be continuing the work that Zev is doing. And this is with the help of Professor Mao, who will be taking over the responsibilities of Zev's current grant and moving the research forward. We're also delighted to have two distinguished speakers with us, and I'll just say their names very briefly now. They will be introduced formally a little bit later in the program, but we are thrilled that Professor Max Cooper and Professor Roy Mariuzza were able to undertake to give seminars to us that will really highlight some of the latest advances in the field of research that Zev worked in and some of his contributions to that field. So I'm going to just lead you through some of Zev's academic background and say a few brief words and then hand over to Yoni Zohar to continue with some reflections on Zev and to introduce our first speaker. So to tell you a little bit about Zev's academic background. Zev did his Bachelor's of Science in Biology at Tel Aviv University in Israel. He then did a Master's of Science in Animal Sciences. He got that degree cum laude at the Hebrew University of Jerusalem in Israel. He then proceeded from 1991 to 1994 to do a Doctor in Science, a DSc degree in Comparative Immunology at the Technikon Israel Institute for Technology. And his advisors there were Drs. Harriet Gershon and Baruch Rinkovich. Now, for those of you who know the universities in Israel, this really means that Zev attended, I think, what are generally regarded as the three very top academic institutions in Israel: Tel Aviv University, the Hebrew University, and the Technikon. Zev then went to yet another highly prestigious institute in Israel for his first postdoc. That was at the Weizmann Institute of Science, where his mentor was Dr. David Wallach. He then moved to Germany and worked with a good friend of mine who studies marine sponges, Professor Werner Muller, and did a postdoc with Werner at Mainz University. At that point in his career, he moved to the United States and worked at the California Institute of Technology with Eric Davidson, and then following that at the University of Alabama in Birmingham with his mentor, Professor Max Cooper, who we'll be hearing from later today. So that's the academic progression that Zev went through before he joined us here. I think it's worth pointing out I had some fascinating conversations with Zev at different times about some of his other jobs that he did along the way. Zev also worked at the Tel Aviv Aquarium as an aquarist for a long time, and he worked as a fisherman for a while as well. So I think you see the pattern of this very close link with the marine environment all the way through his life. So Zev joined us here at the Center of Marine Biotechnology back then in 2005, and he was recruited by Yoni Zohar, who was serving as the director of the Center of Marine Biotechnology, and Yoni will share some of his recollections shortly. In 2010, he was promoted to associate professor, and that was during our transition to become the Institute of Marine and Environmental Technology. So his promotion to associate professor was at the University of Maryland, Baltimore, at the School of Medicine. That's given you an idea of his professional advancement through his career. I think for me, I miss my conversations with Zev. We would often talk over lunch in the fourth floor lunchroom and in the corridors here at IMET. I also miss his highly penetrating questions at every seminar, and that's something to remember when we hear from our seminar speakers today. Don't be shy about those penetrating questions. And his very direct comments and input at our faculty meetings as well. So when we thought about this event today, we want this to be a celebration of Zev's life and with a focus on the science. And we are convinced that that is what Zev would have wanted. And with that, I'll ask Yoni Zohar to say a few more words and to introduce our first speaker. Yoni.
Thanks, Russell. I'd like to join you, Russell, in welcoming everybody here. Obviously, get together celebrating Zeev's scientific accomplishment, Zeev as a person, as a colleague, as a friend, as a family member. And especially, I would like to say to the family, and we have here, obviously, Gladys Siegel, his aunt, Barbara Rabinovich, another aunt, and Rita, a family member. And Gladys, I knew you very well. I knew you very well as your daughter, Talia, from the Zev stories. He kept talking about you. He was so close to you. So it's nice to be able to meet you. I missed you when you visited here, albeit, in such sad circumstances. And I also, looking at you, I can see the DNA at work.
Not as much as I would like.
Both physically and intellectually, I would say.
Yes, definitely.
So I know how painful, and I'm going to read much of it because it's too moving for me. And so I know how painful this has been for you, and above all to his mother, to your sister. I spoke with her a few times, recently over the phone. What an exceptional, special lady, and I know more about her from you. And she really wanted to be here today, but she thought that in this, under this huge pain, it was too much for her to travel here from Israel. And Satoshi, I also want to join in thanking you as well from coming all the way from Japan just for this, to be here. Zev thought the world of you, and I'm sure that from above, or from the Mediterranean Sea where his ashes were spread, so Zev went back to the sea, he would be smiling now, knowing that you are here. So yeah, as Russell mentioned, I was the person who hired Zev. As such, I had very intense interactions with him, which then developed into a warm collegiality and a friendship. Years of talking about science, about life, about biology, and beyond, everything. And obviously, like Russell said, I've been missing this a lot. Actually, this is where Zev was sitting every single seminar and asking all this tough questions.
Look out for my questions.
This is where he was sitting. Okay, since Zev actually passed away, I checked the hundreds of emails I had with him over the years. And the first time Zev wrote to me was in the summer, in July of 1999. This is around where his father passed away. he was then working on sponge immunology with Eric Davidson at Caltech. He already then inquired about the possibility of joining us here. He really loved this place and he really wanted to join us here. Gladys, I told you, I think part of it was, I mean, science was everything for him, but I mean, I think he wanted to be close to you as well. I mean, that's my interpretation. He never said it. He, in that first mail, he reminded me that many years ago we were introduced by a mutual good friend of us, Salome, in Israel, who Zev worked as a fisherman and as a scuba diver in the Red Sea. Russell mentioned it, Zev loved the ocean. And just reading this email in July 1999, I was immediately attracted to Zev. Because this combination of a fisherman and a top scientist, of a sea and an animal lover and a genius of their genes, it's kind of a combination that I knew well had to be here. Actually, about animal loving and so on, animal plants, you know, he was also growing orchids and adamant, you know, orchid grower. But the other day, Gladys, I mean, you told me the story that as a child, Zev chased and played with animals all the time. And you told me the story when he was 16 years old, he actually was making decided to make some pocket money out of milking some very venomous snakes and selling, he was 16 then, and selling them to hospital labs.
Vipers.
Wow, I'll get there. So it's so much like Zev. So he caught viper snakes, kept them until they were really fully loaded with venom, and then milked them. And one day, as he was getting ready to milk one of these fully loaded viper snake, he was bitten by it. He didn't think for a minute. He took a friend of him, took him to the hospital, right, on a bike. He didn't tell anything to his parents, and his parents only later were were contacted by some hysterical nurses at the hospital, begging them to come pick Zev out of there because he was doing all these wheelchair races and all this. They wanted him out of there. So this is so much like Zev. So Zev and I kept in touch, you know, over the years, and I knew that we should do our best to bring him here. And the opportunity came in 2004 when we had an open faculty line in the field of marine immunology. We received multiple very competitive applications, but it was very clear to the search committee, and actually Frank Robb was the chair of that search committee, and Geraldo Vasta and Martin Flenig, and I think Tom, and I think Feng, you were on his committee as well. And I don't remember who else. You were, right? Yeah. And so it was very clear to the search committee and to all the faculty members and that we should hurry in hiring Zev, as he was also courted by institutions like Woods Hole and Berkeley. And obviously I know that Dr. Cooper, you tried to keep him down at the University of Alabama at Birmingham. We offered Zev the job in April 2005, and a month later he was here. And this is what Zev wrote to me when we offered him the job. Quote, "I am very proud to call COM," this was our name before, when Zev was hired, I am very proud to call COM my new home, and I look forward to a spectacular career in my field and as part of the joint effort in marine biotechnology at COM. Apparently, he was really happy to be joining us after all these years and to start his own career. And we were happy too. I can quote you many faculty members' emails about, you know, how great it was that Zev was going to join us. And he made us a better place. There's no doubt about it. Zev's main focus in life, and we all know it, was his science. When we made him, or I made him an offer, he did not negotiate for a second regarding his salary, his academic rank, moving expenses, didn't matter a bit to him. Yeah, no, that's good. He just wanted to know that he had the adequate resources to build a state-of-the-art lab so he can do the best possible science, and he really loved it here. Indeed, joining here, he dove into his science, worked around the clock, and had an extraordinary career here over the years. A few years after joining us, he was promoted with flying colors to associate professor with tenure. I want to take a few minutes to read just a few of the many very telling quotes from his reference letters written by the leaders in his field. And by the way, I did a mistake, I'm sorry. I mean, Frank, you were the chair of his P&T committee, right? And Gerardo, I think you were the chair of the search committee, right? Sorry for the mistake. Anyway, so I'm going to take a few minutes to read those quotes. And again, this is written by, and I'm not going to name them, but the leaders in his field. Quote, "His contribution to our understanding of the evolution of the immune system is simply outstanding," wrote one. Quote, "The sheer joy of having a legitimate excuse to take the time to read Zeev's body of work has been terrific. He is a superstar. Treat him well. The recruiters are at the gate," wrote another. His quote, "The discovery of VLRs by Zev was a big surprise and changed the way how we view the evolution of adaptive immunity," wrote another very well-recognized person, scientist in that field. I think I jumped one. Jump one. His discovery of the VLR genes published in Nature was truly paradigm shifting in that it demonstrated the independent evolution of a second system of adaptive immune system. So paradigm changing, this is a word we hear a lot about Zeev's research. And one of our own faculty wrote, quote, "His finding of humoral immune system that is analogous to antibodies will open avenues for biomedical research in ways that we have not imagined." And this is what Zev was very successfully doing until the day he passed away, combining the most cutting-edge basic research in lamprey and hagfish immunology with biotechnological applications towards improving medicine. This is what he has been doing until a couple of weeks ago. I tried to think about some words describing Zev, and here are some out of many that came up. Authentic, direct, honest, sometimes painfully so. Extremely smart and bright, actually a genius, as told me his PhD advisor, Buki Renkevich. Buki said he was absolutely a genius. Determined. I mean, he didn't think that anything was impossible in science. Motivated, courageous, intellectual, funny, very sensitive, and a good and a loyal friend, especially, at least to me. We much too precautiously lost a very special person, a superstar scientist, in the peak of an outstanding career, a colleague, and I lost a friend. And now, it is a great pleasure for me to introduce one of Zeev's mentors, Professor Max Cooper from the School of Medicine at Emory University. Professor Cooper is one of the world's most prominent scientists in the field of comparative and evolutionary immunology, with research spanning from animal models such as lampreys to humans, and from genes to developing new medical therapies to treat diseases of the immune system. So no wonder that Zev really wanted to go to your lab and learned so much from you. In your lab, at the time at the University of Alabama at Birmingham, Zev went up the phylogenetic ladder from sponges to primitive vertebrates and made some of his scientific breakthrough in developing the other VLR-based immune system. Dr. Cooper, I really appreciate your willingness to be here today. Despite your very busy schedule, you made yourself completely and absolutely available to join us for this memorial whenever it was convenient for us. I wanted to let you know that Zeev thought very highly of you. We talked about you many times. And as he wrote to me in one of his earlier emails, quote, "Wherever I go, I will continue to collaborate with Max Cooper, making use of his enormous knowledge and interest in the evolution of adaptive immunity." He also told me how hard you tried to keep him at the University of Alabama and how committed you were to helping him find a faculty position of his own and start his own scientific career. So again, thank you very much for being here today.
Thank you very much, Johnny. I'm sad but delighted to be here to celebrate Zev Pancer's life. And I'd like to just to briefly set the scene for this, this is a long saga, and I'll just begin at a point around 2000. This search for the roots of our adaptive immune system, at that point, Jan Klein's laboratory had found a transcription factor expressed in lamprey cells in a hematopoietic tissue in what looked like a lymphocyte. There had been earlier evidence that our adaptive immune system went back this far. We knew a lot about the adaptive immune system in jawed vertebrates through studies of pioneers like Robert Good, like Martin Flanagan, his mentor Louis Pasquier, Gary Littman, Bill Hildman, and others, that all of the jawed vertebrates, all of us had basically the same kind of adaptive immune system. So at that time, the search was to go back and to another vertebrate lineage, as you heard, lampreys and hagfish, to see if they had the cardinal elements of our adaptive immune system. And there are only two species that are more basal than the jawed vertebrates. The jawless vertebrates, hagfish and lampreys, they are about 40-something species of each, and they're worldwide in their distribution. Our studies were done mainly with sea lampreys that have a complicated lifespan. They're predatory as adults and can kill about 40 kilograms of fish in a year. And when they made their way into the landlocked lakes of North America, they wiped out native fish. Now this is the hematopoietic tissue cutting a section across one of these lampreys, sea lampreys in a larval stage. That's the site where Jan Klein's group had found by in situ hybridization a little round cell that expressed a transcription factor, 5B, that was known to be important in lymphopoiesis, and particularly B lymphopoiesis. So Jan Klein's lab and our lab began to work together, and the strategy was to take cells from that hematopoietic tissue. It had the light scatter characteristics of lymphocytes that looked like lymphocytes even by transmission EM to make a cDNA library using these cells and then getting sequences, but then with high throughput, to then annotating our catch by the existing databases. We found lots of genes that our lymphocytes use for migration, for activation, and so forth. We found some that were involved in triggering our B lymphocytes, this BCAP and SIG elements of signaling pathways, but we didn't find any of the cardinal elements of the adaptive immune system that we were looking for. And it was about this time that, thanks to Mark Planck, Zev and I began to work together. John Klein was then closing his Max Planck Institute in Tübingen. Martin realized what some of my deficits in molecular biology and Zev's strengths and interests, and thought we might work well together. And that's how my association with Zev began. So, and this is the time that you can see Zev's intent gaze as he did for everything you've heard before. Zev very quickly made another cDNA library. We got another 9,000 sequences, and Zev found two elements, one of which looked like our T-cell receptor, another that was a CD4, a co-receptor molecule ortholog that our T-cells have. And you can show that this TCR-like threaded onto a mouse T-cell receptor B alpha, B region, J region, and found Z and J like regions in this molecule. And so, and similarly, although less well, the CD4-like. You can also show in this virtual northern that the TCR-like was expressed in tissues that looked, that had lymphocytes in them and was not expressed by other types of cells in these tissues, and tissues that were not very abundant in the lymphocytes. CD4 pattern is a little more complicated. So it looked as if in this lamprey there might be these CD4-like T-cell receptor-like molecules. However, Zev could only find a single gene copy of this gene, and everything was encoded in one exon, the V and the J-like regions in there. So it was clear this was not a basis for a diverse antigen recognition system. Not shown here too, it had a long cytoplasmic tail and had inhibitory motifs for signaling in it. So we, at that time, were scratching our heads because we still hadn't found any of the cardinal elements of our adaptive immune system. Now, we knew at the time from studies of many others done much earlier, still that there must be some kind of specificity, and in particular because others had shown that you could make specific agglutinins for heterologous red blood cells, like our O-positive blood cells. And that they had cells that were thought to be lymphocytes that could respond to mitogens and antigens in culture. You hear about this 5E transcription factor. And they were sensitive to steroids, like all lymphocytes are. So Zev and I thought if we could go back and stimulate the lamprey larvae, maybe we could catch the cells in action. And so Zev injected lamprey larvae with a couple of plant mitogens, PHA, pokeweed mitogen, live E. coli, and heterologous sheep erythrocytes. And you can see that now, instead of just this button of small cells by forward and side scatter, there was a vast increase in the number of cells with light scatter characteristics of lymphoblasts, and indeed they look like lymphoblasts. And Zev made a a selective library to look at it was enriched with transcripts made by these cells. And then we started looking through them. We were disappointed, actually, to find that we had enriched for leucine-rich repeat sequences. And this was the summertime, and Jill Settman was home from University of Michigan, and came to our lab because she wanted to learn some molecular biology. So Zev took her under his wing to teach her some molecular biology, and gave her the issue of sequencing these leucine-rich repeats. He knew at that time, having found hundreds of leucine-rich repeats in sea urchins, this was, who had no evidence of an adaptive immune response as the kind that we were looking for. This was probably not what we were looking for. We were disappointed. But at the end of the summer, when he went over the sequences and we had her presentation before we went back, looking at these sequences, it was obvious that every one was different from the other. They had different numbers of these leucine-rich repeat building blocks, and even the ones they shared were different one from the other. And the penny finally dropped that this was not what we thought we were looking for, but this was the kind of diversity that could allow a specific recognition system. And that was a key moment. This is sort of a stick model that Zev made of how these the longest one of those sequences that I showed you before. They're conventional, have capping N-terminal and C-terminal LRRs that they protect the hydrophobic core. They all had an LRR1 and connecting peptide. These green blocks were the ones that were varied. This is the invariant stalk region and hydrophobic tail. Zev made a model using the Swiss model system of this sequence, and you can see that it's just a crescent-shaped molecule with these beta sheets on the palm, and there's some things that didn't fit for this kind of loop. And this is what he learned in the first 112 BLR clones that he sequenced. These came from lots of different lampreys, and every one, as you can see in this phylogenetic tree, almost is different from the other. There's an occasional duplication, and those only came from immunized or stimulated larvae. There's another point that in these stars or asterisks are single cell PCRs that Zev did, and they showed that only one allele was expressed. And we were curious about what was the coding, where did these come from, what were the genes? I'd already mentioned that Zev had found hundreds of LRR in the sea urchin, and plants also have resistance genes with lots of different leucine-rich repeat sequences, and all of them, each one is coded by a separate gene. So we needed to look at the genome, and we knew that Chris Amamiya in Washington had a genomic library made from a sperm from a single lamprey. And so we worked together with Chris in his laboratory, and using probes from that, from the BLR, the ends of the BLR sequences, the N-terminal and C-terminal, you can see from this simple pattern at a glance that there are not lots of different genes. There's only one or two in this pattern. And when we then got genomic sequence, we can only find one gene in Chris's genomic library, and it was very incomplete. It only encoded for the ends, pieces of the ends of the molecule, and this invariant stalk region. And it took us a while to get working with several companies. This was not an easy task to get sequences at that point. There are lots of repetitive sequences. It's still quite a task to sequence the Lamprey genome, but we found, and in analyzing the sequences that we had, Zev could see that there were flanking sequences that could, if they were somehow stitched in to this incomplete sequence, and these large stretches of non-coding intervening sequences could give rise to a mature VLR sequence of the type that we had so many at that time. And it turned out that if we isolated lymphocytes from erythrocytes, that assembly process, or the product that would be shorter because of the elimination of these long tracks of intervening sequences, only happened in the lymphocytes, not in the erythrocytes. Zev cloned and sequenced lots of these, and they too were all different, one from the other. Now, this is that report that Yanni mentioned in Nature earlier, and this is a diagram stolen from a News and Views that Martin Flanagan wrote about this article. That synthesizes what we were thinking at that time that was might be the situation. So here's the Agnathan, the Lamprey incomplete genome with use of these somehow inserted in here to generate cell surface molecules, and we could show that that recombinant molecule of one of these sequences that we had expressed on the surface and could be cleaved off by a phospholipase C of bacterial origin. So we assumed that they would also be secreted in a multimeric form, at least a dimeric form, in order to agglutinate things like red blood cells that they were seen. So we know more about this process now, but we still don't know the full sequence. But we know we had captured some of these intervening sequences, and that was done in our laboratory by a PhD student, Matt Alder, again working with Zev. And Nagata and their colleagues showed that this process could not only begin on one end or the other, but it never happened on both ends. It's a very controlled process. We know that based on short stretches of nucleotide sequences with the donor and the recipient, this is brought in and the sequence begins to be copied in for the next step. And this stepwise, piecewise process goes through to completion, beginning on one end or the other. So are these the things that are causing the agglutinin responses that have been reported much earlier? And I mentioned we've used a lot O-positive erythrocytes that have this characteristic H-trisaccharide as a dominant antigen, and a BCLA molecule, a hair-like nap on Anthrax exosporium. These are small creatures, as you can tell by the size of the fingers in this insulin syringe to inject the antigen into the coelomic cavity, which is comparable to our peritoneal cavity. And just as had been reported by others, we could show that after one immunization, these larvae after a couple of weeks made agglutinins, and after a booster immunization, 10 to 100 times higher levels of the agglutinin. What I didn't tell you is that that invariant stalk allowed us to make a monoclonal antibody against these VLRs, we call them, or variable lymphocyte receptors, and that removed all of these. So that was what was made. These are the cells that Matt Alder could show. Here you're looking at a cross-section of the gills of a lamprey larvae, and these green cells are the ones that are making the variable lymphocyte receptors, B, we call them. And before and after immunization, these yellow cells are the ones that proliferate in response to the immunization. The cells that are secreting those antibodies, or VLRs, look like plasma cells or even plasma cells. And the Rand could show that these are very large molecules, except when he reduced disulfide bonds, they fell apart into smaller units. It was not possible to look at the structure of this mixture, so Rand devised a way to make monoclonals by preparing messenger RNA, constructing a cDNA library, and expressing them in human embryonic kidney cells one by one, and then looking at the antibodies, and that allowed a look with negative image EM to see that these look sort of like shown in this cartoon. The antigen binding parts are in these leucine-rich repeat part of the structure that I mentioned, and that could be shown by mutating one of these different residues in this coding regions for different residues to increase or decrease the binding of it. The C-terminal stalk has these cysteines, and I did mention that allows them to be bonded. And one of the structure branch antibodies, this one against sheepherid, the H trisaccharide of O-positive human blood cells, could be shown here. And I mention it because Zev had noted these are the binding sites, these are hydrogen bonds. Here you'll hear more about from Roy's much deeper presentation. The point I wanted to make, though, is that Zev had noted in this C-terminal LRR that there was a highly variable insert. Turns out that encodes this loop that's sticking out here, sort of like a thumb in the palm of a bent hand, and that's involved in the binding. So this allowed us to know that there was a gene that was assembled in at least partially depicted ways here, and in a pathway of differentiation that looked like our B lymphocytes. And I thought, aha, that I always thought B cells would come before T cells because it's a simpler, easier thing. You don't have to process the antigen, present it, and so on and so forth. However, the story is more complicated than that, and Zev had worked with Kasahara. Masanori Kasahara had a large cDNA library, and we contacted him, and by sifting through his cDNA sequences could find two hagfish genes. They were both partial, and sequence around them indicated that they had flanking sequences that allowed them to be assembled into mature VLRs like this. So they were named VLR-As and VLR-Bs. Now, later on, Zev was able to show, after he moved here, he and his colleagues found a second VLR gene. And moreover, with Igor Rogozin, who's here today, and they and their colleagues identified two AID-like organoids in lamprey that they call cytosine deaminase 1, CD8-1, and CD8-2, that had resemblance to our AID molecules that are involved in generation of diversity by gene conversion mechanism, birds and some mammals like sheep and rabbits, and also they're involved in somatic hypermutation and class switching in versions. Now we know a third one that Kaspar's group found. So lampreys have 3 of these genes. They're all incomplete. And moreover, they're expressed by separate lymphocyte lineages, 2 of which look much like our T-cell lineages. They respond to antigen, to phytohemagglutinin like PHA, but they don't secrete their receptors. And they are generated, we've been able to find what Thomas Boone noted, in a thymus-like equivalent. And moreover, the CDA1 was important in identifying where they're generated, and CDA2 is only expressed by the BLRB lineage cell. They have a similar pattern of at least a basic genes that our T cells, alpha, beta, and gamma delta like, that our T cells make, and that our B cells use for commitment to the pathway. Here's an example of that CDA1 is only expressed in the tips of these, this is the cross-section of a fold, a gill fold, and you can see that that's the only place that this cytosine deaminase is expressed, and this first of the secondary follicles here. So it's a complete surprise to that it looks as if the basic genomic program is for the lymphocyte pathways, the major one that we have is already existent in a common ancestor of Jawless and Jawless turtles. The hagfish and the lamprey now have been shown to have 3 BLR genes. So this little cartoon just revamps how we begin to look. So hagfish and lampreys had a common ancestor about 480 million years ago, and they have these BLR, A, B, and C genes and the lineages. The common ancestor of ours, somewhere around half a billion years ago, must have had the genetic program for these basic fundamental pattern for lymphocyte differentiation of a primordial T cell, gamma delta like, alpha beta like, and B cell like lineage. And you'll be hearing much more from the many contributions that Roy Mariuzza has made about the details of these antibodies and why he and Zev, we think that these are going to be biomedical important. Zev, there are a lot of people who've contributed to this story that's certainly reshaped our view of how adaptive immune systems might evolve. And hopefully will have some medical potential. And I've mentioned many of these along the way. Everyone in our group joins me in paying tribute, celebrating Zev's life, and I'd like to just close with, it's hard to think of something that would be exactly appropriate for Zev. He was such an exceptional person. But these words that I found sort of somehow seemed appropriate to me. A single field which I have looked upon, both of them speak of something that is gone. Pansy at my feet doth the same tale repeat. Whither is fled the visionary gleam? Where is it now, the glory and the dream? Thank you.
Thank you so much. Now we can open it for a couple of minutes of small discussion if anybody wanted to ask or to comment.
Where is that?
He will be the first, and he and I will
That's
always
exactly right. So, why three types? That lineage is very, very interesting. The fact that there's replicated. Is that
Alan, I don't know.
No, I'd love that.
It's one of the questions, but it must have some fitness value or it wouldn't have survived, I would imagine. I don't know what the receptors were before the convergent evolution of these two types of highly variable types of receptors. One thing we've learned is that the DLRA and the DLRC genes are close together.
Okay.
And they are expressed at about the same time, perhaps C a little before the A, at a time that the B, which is quite distant, is silent. And they even can share some of the donor sequences in their assembly. And that's, again, another sort of think-alike, at least, of our gamma delta and alpha beta T cells, which the delta locus is in the middle of the alpha gene locus, so they can use some of the same variable genes. What that fitness value is, when we find out what they actually see, and what allo determines that these creatures, A and C sod-like cells, preferentially respond to, then I think we'll creep closer to an answer to your question.
Actually, you know, remembering Zev, I mean, he never would wait until you to finish a lecture before he asked his question, right? So he had a question, he would stop you and ask you right there when, you know, he had it.
Now, relative to his sensitive nature, it's really pertinent when he put lamprey on trout or salmon and had to rear them.
Right.
That was one reason why I don't think he wanted to continue.
The animals.
Right.
But, which reminds me, another thing, you know, when he came here, because my expertise is like closing life cycles and, you know, spawning, he wanted to close a life cycle of this lamprey is right down here, so it drove me nuts. But bringing those animals, big animals here, and now starting to feed them, right? With salmon and trout. So
that
for
really,
a while,
really
work on this together. But yeah, I think it was too much for me.
Yep.
Do you have an idea of Zev's vision that drove all this intense research? What he was hoping to achieve with all this? This is intense. Not being a scientist, a linguist.
I think it was curiosity. He was an intensely curious person. He was intensely focused. Jonathan and I were talking this morning. Every day he would come in to work, he had formulated an idea or a question that he had been arguing with himself through the night and confront you with this question. and there was no changing his mind because he had thought it through in a very deep way and was convinced that he had reached the probably the right conclusion. He just wanted you to parry with him a bit.
And he was at the cutting edge, you know, so really, I mean, just making sure that there is progress in the most advanced scientific research, just a better understanding of our life. Anyway, thank you very much, and I'd like to introduce now another good friend, colleague, and somebody who really understands as well, better than me, the contributions of the Professor Martin Fleming from the School of Medicine.
I'd like to, I also have to introduce Roy Mariuzza to give the second talk, and I'd like to do that before I give my little tribute, because I don't know if I'll be able to at the end. It's really a pleasure to have Roy Mariuzza here to give the second talk. Roy worked with Zev, as he'll tell you, and Roy is part of the University of Maryland system. He's worked at CART for the last 20 years. He's been someone studying crystal structure of immune proteins for 30 years. For the first 10 years, mainly at the Institut Pasteur and in Cambridge, and then moved to CART about 20 years ago. And he's one of the handful of structural people in immunology who have contributed so much to this field. And on top of that, he's such a wonderful guy. Actually, one of his proteges is now a faculty member at the IHV right down the road, Eric Sundberg, who is also a wonderful person doing great things. And if you want to get a really good snapshot of the work that Roy has done over the past 10 years or so, Frontiers in Immunology just came out last week, a review article on the work on antigen receptors, natural killer cell receptors, major histocompatibility complex, where I think encapsulates his work on at least the jawed vertebrate system. So when I was writing this, or thinking about this tribute, surprisingly, I was thinking of the Gettysburg Address, strangely enough. Maybe it isn't so strange because I think the world of Abraham Lincoln. And over the past couple of years, there have been great books that have come out on Lincoln, and of course, the movie was fantastic. And to start off this Zev Pancer Memorial Symposium, I think it's altogether right and fitting that we should do this as part of the Gettysburg Address. So I met Zev first about 20 years ago, and it's been said, and it seems to be true, he was an intense fellow, you could say. And he did a postdoc with Eric Davidson in the early 2000s, and a close colleague of him, Jonathan Rast, was doing a postdoc at the same time. And Jonathan had bookended two things. So he had something to do on Thursday, something to do on Saturday, drove all the way down from Toronto and back, and he was going to attend both of those things, but he had to be here for Zev's symposium. And we're really grateful that he came down. In 2002, Zev finished his postdoc at Caltech, and he was doing a road trip looking for a job. And as part of that, he came to Baltimore, and he was thinking of working with me. Fortunately, I didn't have any money at the time, and I wish I could have hired him. I'm sure we would have done some really interesting things together. But since I didn't have money, I knew that Max had just come out with a couple of papers in PNAS, and they identified, as he told you, a way of isolating lymphocytes just by light scatter. His long experience in jawed vertebrates gave him the expertise where he could do that in the lampreys. And the first papers that came out looked at ESTs, and honestly, Max, it didn't look that interesting to me. There was no IG, no TCR, no MHC. A lot of us had tried to do that for a long time. A lot of student and postdoc careers went down the tubes trying to find IG, TCR, and MHC. But still, I knew Zev, with his molecular expertise, if he went to Max's lab, that it was a match made in heaven, and something good was going to come out of that. Yeah, really, something good came out of that, way beyond any of our expectations, or none of us had any idea that there would be some type of convergent adaptive immune system in the jawless vertebrates. So we were thrilled when Zev came to Cone. And when he came here, he and I had one try at a collaboration, and it didn't work out for whatever reason. A couple of years later, Zev called me up and said, "Martin, I have adult lamprey. And would you come over and help me immunize and feed them?" That would be great. And I jumped at the chance because I love working with animals, and it was really cool because all the work that had been done until that time had been done with the larval lampreys. And as you just heard a minute ago, these were the adult lampreys, which had to be fed on bony fish. So I'd come over, and we would immunize the animals and bleed the animals, and I thought it was fantastic. We'd open up the tank, And there were the lampreys feeding on those bony fish, and it was a gory beauty to me. I just loved watching that. I think, you know, I really don't like Telios fish. I think they're overrated. But that was the first time that you know, I saw this compassionate side of Zev. He absolutely hated this. And you know, I was really taken aback. I would joke about it at first, but you know, you don't joke with Zev about something that he's really wrong about. So I stopped doing that. And anyway, this was a wonderful collaboration. This is where Roy stepped in as well, and fantastic results came out of those two papers. So much so that it opened up all these other questions. And you know, I begged Zev. I said, Zev, you know, we have to write a grant on this to follow it up. I did that several times, and probably some of you know that when Zev makes up his mind, I mean, you could beg on your knees for hours, and it's not going to change his mind. You know, if he wants to go right and you want him to veer a little bit to, you know, semi-right, it's not going to happen. He's going right. And he decided at that time that he wasn't going to study physiology anymore. He wanted to use these lamprey antibodies for therapeutic and diagnostic reasons. And actually, I kept trying. I'm a persistent guy, I have to say. Up until there were these joint IMET UMB grants that we had a possibility of writing, and I would bug him every week to do it. And no, I see no reason to do that. This just doesn't work. NIH will never fund this. Okay. Anyway, this you know, strong personality of what I'll do, what I won't do. I think it didn't serve him right sometimes, because there were things that we could have done together. On the other hand, I've never seen anybody like this who was able to take on a project and have tunnel vision and not be blocked in doing that project. Nobody did I ever see like that. And of course, that served him so well in these fantastic discoveries that he made. So getting back to the Gettysburg Address, remember Lincoln said, the world will little remember what we said here, but it can never forget what they did here. Okay. So this symposium, a couple years from now, we're not going to remember it. Okay. But this finding of this convergent lamprey adaptive immune system, as long as there's immunology, this is going to be in the textbooks. Okay. You know, until the meteorites hit us. And this is something Zev found, and there aren't many of us who can say that we're going to live forever with our discovery. And to finish, I've had this Don McLean song, Starry, Starry Night, just going through my head on a loop for the past month or so. And, the last sentence of that song is, but I could have told you, Zev Pancer, this world was never meant for one as beautiful as you.
Met Zev actually in 2006, soon after he came to Maryland, and it was at the occasion of a seminar that he gave here, and he presented his VLR work, and I thought it was the most fantastic thing. I was really amazed It was just so novel, so original. And I thought, well, maybe we should collaborate. And since I was interested in the structure of immune system receptors, this could be a really productive, complementary type of collaboration. So after the seminar, I went up to Zev and I introduced myself. And I proposed maybe we should look at the structure of these things together. So he was quite skeptical, and he asked a lot of tough questions. It was a difficult interview. And that's how I first learned how passionate and committed Zev was to his science. So I didn't hear anything for a few days after that, and I thought, well, that's probably the end of it. But then he called up and said, okay, so I've thought about what you've proposed to do, and I think we can try to do this together. And so we did. And everything I'll tell you about today is work that we did in very close collaboration. All the molecules I'll talk about are molecules that Zev isolated here at this center. So my interest is in antigen recognition by the immune system, and the work I'll tell you about, of course, has to do with the variable lymphocyte receptors that were identified by Max and Zev about 10 years ago now. Okay, so as Max told you about, if you look at this animal tree of life, all right, everybody has an innate immune system consisting of knobs and toll-like receptors and so forth, but only the jawed and jawless vertebrates have an adaptive immune system. And of course we know lots and lots about the receptors of the jawed vertebrates, but even now, not that much about the jawless vertebrates. This is of course from a review that Max and Zev wrote soon after their really classical discoveries. So, okay, so we thought we'd like to look at recognition by these jawless vertebrate receptors. And well, this is an example of a lamprey that Zev and Max studied. This is an adult lamprey. It's quite large, it can grow to several feet. Okay, so the real breakthrough discovery, which Max told you about, is when Zev, in this classical paper, described how these lamprey lymphocyte receptors, these VLRs, are assembled from these leucine-rich repeat cassettes. So up until that time, everybody had assumed that the adaptive immune system of the lamprey would be pretty much like ours, based on immunoglobulin domains, but that of course proved to be completely wrong insofar as they're assembled from completely different protein scaffolds. So that was really a groundbreaking discovery in the field of immunology. All right, so we have these two, vertebrates have these two types of adaptive immune receptors, Ig-based or leucine-rich repeat-based. Okay, and as Max showed you, these are 20 or so random VLR sequences to show you how diverse they are. And some of them are quite long, 200 amino acids, some of them are much shorter, 120 or so amino acids. But the point is that there's a lot of diversity, and there's as much diversity in the VLR system as there is in the antibody or T-cell receptor system. Well, okay, so Zev was an evolutionary immunologist, and we know a lot about how the jaw vertebrate receptors recognize their antigens. Here's an antigen-antibody complex. Here is a T-cell receptor recognizing peptide MHC. Okay, so the antibodies, they bind antigens directly, and T-cell receptors, or at least alpha-beta T-cell receptors, they only recognize peptides bound to MHC molecules. So the antibodies recognize native antigens, and the T cell receptors recognize processed antigens. So our interest was to see how the VLR recognition system compares with this IG-based system. Okay, so that was quite a challenge. And the challenge really was, how do we get monoclonal VLRs? All right, so we didn't have a hybridomal system, as in the case of antibodies. We didn't have a phage display system either. So Zev, he realized that we needed a method for pulling out antigen-specific VLRs from library. So what he did with Satoshi Asumi, who's here, okay? Satoshi, okay? So they developed this very innovative display system for isolating monoclonal VLRs, essentially. Okay, so the first step was to choose a model system, model antigen system, and we chose lysozyme for reasons I'll hope to make clear shortly. Anyway, Zev, with Martin Flanagan's help, right, he showed that lampreys could be immunized with this small protein, and there could be an immune response, which could be quite strong, right? Okay, so that was fine, but then how do you pull out specific VLRs from the millions of VLRs in the lamprey? So that's when Zev and Satoshi developed this yeast display system. So basically what they did was to immunize the larva of lamprey, and then to make libraries from the messenger RNAs encoding these VLRs, and then to display these VLRs on the surface of yeast fused to a yeast surface protein. So that way you could have each yeast cell expressing an individual VLR, and you could then screen this library by flow cytometry using a bioattenuated and fluorescently labeled antigen, in this case lysozyme. So this was actually quite an achievement to put into place. I mean, Zev tried other expression systems. He tried phage display, but that did not work. And he actually developed this new method for anchoring VLRs to the yeast surface. There had been other approaches which were successful with antibodies, but for VLRs, he found this other type of anchoring strategy which worked quite well. So with that technology, Zev then proceeded to isolate monoclonal VLRs from these libraries. And at that point, okay, we were able to begin our collaboration in characterizing these molecules at the protein level. So this is an example of one of these VLR clones pulled out from the library, which recognizes lysozyme. So this is an affinity measurement, essentially, for how this monoclonal VLR binds lysozyme. And we came up with affinities in the micromolar range. So this was quite interesting because we could not get higher affinities from this system. They all seem to fall in the micromolar range, but actually these affinities were similar to those of IgM antibodies, which are the first antibodies to appear when we get immunized with an antigen. And what was interesting also is that, as Max showed you, these VLRs are not monomeric in plasma, they're multimers, just like IgMs. And what's apparently going on is that VLRBs, they overcome their low monomeric affinities for antigens by forming these multimers which confer high avidity. So this is an example of a convergent strategy to achieve high affinity, high avidity, let's say, from low affinity interactions which is also present in our IgMs versus our IgGs. Okay, so then we had a starting point for looking at these molecules in more detail. And this is the first paper that we co-authored with Zev. And every paper or every piece of data I'll show you actually was all done in collaboration with Zev. And so in this case, we have a complex between the lysozyme molecule and this particular VLR. All right, and you can see that the VLR is contacting the lysozyme through a combination of interactions with these beta strands corresponding to the leucine-rich repeats and this loop, which is present in the C-terminal capping module. And if we look at this from another angle, this cleft is the active site of the lysozyme molecule, and this loop is projecting into the active site cleft. So we can compare that structure with another structure which Max showed you, which was done in collaboration between Max and Ian Wilson's lab, in which another VLR, RBC36, was complex to this trisaccharide antigen here. And that structure is overlaid onto the lysozyme structure here. So here the sugar is in a pocket between the beta strands of the concave surface and this loop here. And in the case of the protein antigen, there are additional contacts simply because the protein is larger. But a really unique characteristic of these VLRs is in fact this loop here. And it's actually almost exclusive to the VLRs among all known leucine-rich repeat proteins. So this is an alignment of leucine-rich repeat proteins of different sorts. And this is an insert which is found in the VLRs, but is not found in toll-like receptors or no-go-like receptors or any of these. It is, however, found in this GPI B alpha protein, which is a platelet glycoprotein receptor, which may be a precursor of the VLRs, an evolutionary precursor of the VLRs. All right, so the reason that Zev and I chose lysozyme to work with is because there's lots of structures of antibodies complexed with lysozyme. And we were interested in seeing, well, are there parallels in the way that lysozyme could be recognized by VLRs versus our antibodies. And this is our structure here on the left. And these are two other structures. One is of a camel antibody, shown here, and one is of a shark antibody, shown here. And the lysozyme molecule is oriented the same way in all these structures. And what you can see is that in each case, especially here, in the case of the camel antibody, the same epitope on the antigen is being targeted. So here you have a case of two completely unrelated antigen receptors recognizing essentially the same site or epitope on this target antigen, which tells us that there's probably something physico-chemically favorable about this binding site, probably at least in part because it is a cleft and it's something which is easier to latch onto than other parts of the protein. And this loop here is the third CDR of this antibody molecule, which is analogous, if you will, to this loop of the VLR. So just to emphasize the similarities between these epitopes, this is the surface of lysozyme, which is contacted by the VLR. This is the surface of the camel antibody, which is, this is the surface of the lysozyme which is contacted by the camel antibody. And what's in green is what's common between these two complexes in terms of what residues on the antigen are contacted, and what's purplish is what's unique to each epitope. So you can see basically there's a lot more green than there is purple. So the epitopes almost completely overlap. And well, we went into detailed studies of the interface, which I won't belabor here. But basically, here we're looking at the interface between the lysozyme down here and VLR up here. And there were a lot of parallels globally between the way VLRs recognize antigens and the way antibodies do. So in terms of the size of the interface, the number of residues involved, the degree of complementarity, very strong similarities to the antibody case, even though the scaffold being used here is completely unrelated to the IG scaffold. And we did structures of free and bound forms of this VLR. So the bound form is shown in orange, and the unbound form is shown in yellow, and we were able to detect some differences in the conformation of the VLR as it binds the antigen. So it's almost lock and key, at least in this particular case, but there are adjustments made in the level of side chains and in movements of certain loops. So there is flexibility. All right, so then we looked at the general features of the antigen binding site. So here we're looking at the surface of the VLR, which contacts the lysozyme antigen, and what's highlighted here in color are contacting residues in the crystal structure. And you can see that these residues essentially form, if you will, three parallel ridges, and they correspond to residues on these beta strands. And in addition, there is this loop here, right? So there were quite a lot of VLR sequences becoming available at this time. And so, okay, Zev said, well, let's look at the diversity of these sequences to try to correlate contacting positions with diversity. That is, let's look at this system in the same way that Wu and Cabot had done long ago in the antibody case, which allowed them to identify CDRs, hypervariable loops. So when we did that in collaboration with El Aravind at the NIH, we saw a very clear picture. Okay, so these are variability plots for the different UC grid repeat modules which compose this VLR. And so if we just look at this module here, on the y-axis is a variability as a function of position along the sequence. And what you can see is that there are four residues which stand out above the others. And that's the case for each of these modules, essentially. So these are the hypervariable residues within the VLR structure. And in these red balls or green triangles, they are residues which actually make contacts of different crystal structures. So what you can see is that these hypervariable residues are also the antigen contacting residues, and they correspond to these ridges here, right? So there's a very close correlation between variability and antigen recognition, as there is in the case of the antibodies. Except that here, these variable residues are actually found on beta strands, whereas in the antibodies they're found on loops. Okay, but there is also a contribution from the loop here, important contribution. There's a variability plot for that. and also contact residues are quite hard. So these VLRs have very modular structure. It's the way perhaps an engineer might think of assembling a recognition system. You line up these short or relatively small components of about 25 amino acids each, different combinations, different lengths, and that's how you build a unique antigen recognition structure. So these VLRs that I told you about so far are just VLR-Bs, and they had relatively low affinities for antigen, about micromolar. So Zev wondered whether it's possible to get higher affinity ones, or whether there's some limitation to the protein scaffold. And so he showed that there's really no limitation in terms of affinity, that you can affinity mature these molecules in vitro and get affinities in the nanomolar range. And that was important for his thinking later when he began to shift more towards the biotechnological aspects of these VLRs. Okay, so about that time, Max and Thomas Bohm, they discovered that there are there are now three, as we just heard, VLR lineages, A, B, and C, right? So I just told you about the Bs, but there's the A's and the C's. And the first to be characterized were the A's. And as Max said, there are very striking functional parallels between the A lymphocytes and T cells, and between the B lymphocytes of the lamprey and our B cells. So everything we had done so far was with Bs. So, but Zev said, okay, let's look at how the A's might recognize antigen. Okay, so maybe they are like our alpha beta T cells that recognize only processed antigen. Maybe they're like gamma delta T cells that might bind antigens directly, or maybe both are possible. So what Zev and Tasumi did, Satoshi, is to go and make a VLR-A library, and from that library they were able to pull out clones which recognized lysozyme. And what was surprising was that the affinity of these clones was really very high. So they went into the nanomolar range, even into the picomolar range. So we still don't know quite what that means. But anyway, and Max and Zev and I, we have some different views on that scientifically. But this is what we pulled out. So we did do the structure of this VLR-A complex with lysozyme. And this is in contrast to this structure here. So this is the high affinity interaction. This VLR happens to be longer than this VLR-B. Okay. It's much more complementary, it probably accounts for the higher affinity. Right. So if we look at the VLR-A epitope on lysozyme, that's shown here. This is the surface of lysozyme. And this is the VLR-B epitope. And what's here are epitopes on lysozyme that are recognized by antibodies, mouse or camel antibodies. There's a lot of overlap between this VLR-B epitope, as you saw, and this camel antibody. But this VLR-A epitope had not been previously detected among the known anti-lysozyme antibodies. So it may be an epitope which is favorable for recognition by VLRs, but less favorable for recognition by antibodies. Okay, so this is perhaps an overview of what we know structurally about antigen recognition in the jaw and the jawless vertebrates. So here we have the antigen antibody system, the alpha beta TCRs recognizing peptide AC, and a gamma delta receptor directly recognizing the antigen. So the VLRBs were like the antibodies. This VLRA may be like the type of direct protein recognition mediated by gamma delta TCRs. And a big question in the field still, and I think people are probably getting closer to an answer, is whether there are presenting elements which may be functionally analogous to MHC molecules. Okay, there's no MHC molecules like molecules in the lamprey, as Max told you. There's nothing that looks like MHC, but then it doesn't need at all to look like MHC because VORs, they don't look like T-cell receptors. So why should it be an MHC-like molecule, except function? All right, so at that point, okay, Zev's thinking began to shift a bit. So he was thinking more in terms of what are the possible biomedical applications of VLRs. He felt that in terms of evolutionary immunology, he would move away from that area more and focus on possible applications. And he first tackled the problem of sugar recognition. So he thought, okay, so to make VLRs viable biomedically, I mean, they have to be able to do something that antibodies don't do or don't do so well, because otherwise, well, why shift from antibodies? And so he chose a very challenging system to look at, which was sugars. So, as immunologists know, it's difficult to make antibodies against sugars, really specific high-affinity antibodies. Part of the reason is that, well, we're self-tolerant to our own sugars, so it's difficult to make antibodies against self-entities. All right, but he thought, well, maybe the VLRs are good for finding reagents to mammalian or human sugars because lampreys are just so evolutionarily distant from the jaw vertebrates, right? And he further thought, well, I don't want to have to immunize at all lampreys because this is a painful process, right? Well, for the investigator and for the lamprey as well. So he said, well, it would be great if we could make generic libraries. Just have a VLR library in a tube against which you could screen whatever target you're interested in and pull out your desired reagent. And so that's what he did. So he made very large, very diverse VLR-B libraries from lampreys from the wild that had never seen any particular antigen. Well, they had seen lots of antigens, but none of the ones that we were going to screen against. So Zev began screening his generic library against all sorts of sugars, some of which are shown here, some disaccharides, monosaccharides, polysaccharides, and he found, I think quite surprisingly, that he could pull out binders against almost any target sugar from this unimmunized library. So here is a binder which recognizes just a monosaccharide nanos. Here are binders that recognize the trisaccharide Lewis A antigen, but it does not recognize X. The reverse here, this one recognizes Lewis X but not Lewis A, and so forth. Okay. So this was very, I think, a striking application of the VLRs. And he chose in particular this antigen here as a test case to prove that VLRs did have potential, biotech potential. And this antigen is a tumor antigen called Thomson-Frieden-Reich antigen, which is just a disaccharide. And he isolated this particular clone and characterized it. So this is a glycan array to look at the specificity of this VLR. So antibodies, they have a problem oftentimes of not being very specific for a specific sugar, but this VLR, when screened against 600 different sugars, really was very specific for this antigen. There were a few cross-reactive sugars, but they were actually structurally related. So this level of specificity is really quite noteworthy. And he showed further, in collaboration with Professor Mao, who's here, that you could use this VLR to specifically stain various lung cancers, which are shown here, but this VLR did not stain normal lung. Okay, so that indicated that these VLRs may have diagnostic potential. And furthermore, these VLRs can stain a significant percentage of lung cancers, and the degree of staining correlated with survival. So the more positively stained the tumor was, the less likely the individual was to survive the lung cancer. Okay. And very importantly, he showed that the avidities of this particular VLR for TF antigen was really quite high. It was actually in the nanomolar range. And that's really critical for any potential diagnostic application or otherwise. So these VLRs turned out to be as good in terms of avidity and specificity as the very best antibodies. And this is a measure of affinity at the monomeric level measured by ITC. So affinity is as good as, for example, this lectin called Griffithsin, which recognizes a diamine. And unfortunately, in our very last paper together, we looked at the interaction of this TF antigen with the VLR to understand the specificity. And this is the structure that we did of that complex. So here is the sugar here in the pocket between this loop and these beta strands. And here is the sugar bound to the VLR, and you can see that there are a number of hydrogen bonds in this interface. And to look at why it's so specific, you need to look at primarily these tryptophan residues. Right? And here we're just highlighting the tryptophan residues. And you can see that they form a type of cage around the antigen. So it's a very tightly fitting hydrophobic cage, which probably explains why not very many other things can fit into this pocket. And these studies really encouraged Zev a lot to go further in this direction of biotech applications. So we had hoped, to continue the characterization of these glycan-specific VLRs and VLR-specific for other difficult antigens. But I hope, that this work can spark, and I'm sure it has, sparked interest in others in the field, and that they'll be able to carry on. And I know Max also is interested in possible biotech applications of the VLRs. So we finish here, and these are the people who I've had the privilege with to collaborate on this work. And really, I think among all the people I've collaborated with down through 30 plus years, Zev was really the most unique collaborator. He was the most passionate scientist I've really ever encountered, I think. He was really deeply committed to his science. He told you exactly what he thought. Things were not always smooth between us, not at all. I can remember quite a few heated arguments, but they were scientific arguments in how to proceed, how best to proceed. And as Martin said, he was very difficult to persuade to go in a direction other than that which he had decided. But fortunately, he had very good judgment and very good foresight. And in the end, I generally went along with what he said. And in retrospect, I don't regret that one bit. So I'm really very sad to have lost Zev as a friend and as a collaborator. And I think it's really a loss to the field. So with that, I think I'll finish. If you have any comments, questions.
Thank you, Roy. Questions for Roy?
Yeah.
He's moved to the biotechnology applications. Can you give us an idea of when chronologically that happened? I mean, obviously the publications that you cited are 2013, but when did you sense that he started moving in that direction?
I think it would be more like 2010. All right. Yeah. It was clear, yeah, that he was already shifting his interest at that point. Yeah.
Oh.
You showed A and B, and is it the character, the blend of those, the beta sheet, is characteristic of higher affinity or not?
No, not necessarily. No, you can have short VLRs with higher affinity than long VLRs. I mean, you have to look at the specific interactions. And actually, depending on the antigen, not all of the leucine rich repeat modules will be making contact anyway. So even though it's longer on the N-terminus, and so those VLRs are not actually contributing. Those leucine rich repeats are not actually contributing necessarily to the interaction.
Well, what I'm actually asking is why make bigger repeats then? Or is it just an artifact of the machinery and insertion?
Oh, why make bigger repeats? Why make bigger VLRs?
Yes.
Oh, because we have to confront, not we, but the laboratory, they have to confront a very diverse energetic universe. So the more diversity you have, presumably the more you can recognize. And also in the case of sugars, so sugars have an elongated structure. And you can imagine, okay, so here we just looked at a disaccharide, which is short, but what about trisaccharides or polysaccharides? So those would presumably extend across the concave surface of the VLR. And so the more modules you have, the more potential contacts you can make with that type of elongated antigen. Yeah.
Have you crystallized VLRCs?
Yeah, but not. Grossly they look like A's and B's, but we don't have any complex with an antigen, so there's not that much we can say about VLRCs at this point.
Let's take one last question from Frank Robb.
So many of the antibodies from vertebrates have been converted or made into catalytic antibodies because of their high affinities. Would you say that there's potential amongst the VLRs for catalytic design? And would this introduce a new aspect to the study of these and their application?
Well, I mean, there have really not been any, in the end, there's not been really any good catalytic antibodies. Okay, this was an area which had a lot of interest 20 years ago, and there was an enormous effort put into isolating catalytic antibodies. And there were antibodies which had some activity, but in no way comparable to enzymes. So I don't think I'm sure Zev was not thinking in terms of catalytic VLRs, at least not that I'm aware.
So please join me in thanking Roy again for an outstanding. We now want to open this up for just a few minutes. If anyone has any short remarks that they would like to make or any recollections in our last few minutes here, please.
I would like to take the opportunity to thank all of you. On behalf of my sister, on behalf of the family, you are, each and every one of you, amazing. I want to highlight especially Satoshi and Sean, who worked shoulder to shoulder in the weeds, 10 hours a day, not easy, all the time. Zev appreciated you and loved you and considered you the most important people in his life. Science wasn't the most important thing to Zev. Science was the only thing that was important to Zev. I benefited. I want to thank you for bringing him to Baltimore, and that he chose to come to Baltimore. For me, it was a gift. I'm down the road in Washington, D.C. So it was a tremendous gift for me to have him these 9 years. And I want to leave you with 2 tidbits about Zev. One is that he adored Zen. Zen gave him comfort, gave him wisdom. Satoshi visited a Zen temple with you when he was in Japan, and he loved it. He just didn't stop talking about it. The other thing is that Ruth, my sister, raised this boy and told me that she considers this the biggest adventure in her life. It wasn't like she could have changed him. She rode the tide with him and loved every minute. Even the day in his early teenage years when he showed up in their apartment in downtown Tel Aviv with a puma on a leash. Why? Because the zoo in Tel Aviv knew of Zev and knew his love of animals. They had a puma cub that had been rejected by its mother. And they called Zev to take care and bottle feed the puma cub. So one day he had to show up at home, and he had the puma, the 6-month-old big puma by this point, on a leash, into the house, into a small elevator with neighbors. So this is the kind of boy that Ruth raised, and the adventure that it was to be his mother, to be his aunt, to be his friend, and to have him. And for that, I'm very grateful to you that you brought him to Baltimore.
Moments to give these to you to take away with you, and thank you so much for being here today.
It's been a pleasure and an honor to be here. Very comforting, certainly very comforting.
Thank you. So any other comments? Dave, did you have some thoughts to share?
Sure. Hi everyone, my name is Zev, and I'm very pleased to be here today. I didn't know Zev as long as some of his scientific colleagues and some of the people who have helped train and worked with him, but I'd say the 2 words that characterized my reaction and my interaction with him were integrity and intensity. So I first met Zev when I was an administrator during the UMBI reorganization, and you heard some aspects about his marvelous career and his ascent, rather rapid ascent, to promotion. And one of the things that happens when people get promoted is they usually get a raise. And one of the things that when I was working with actually Rose Jagus, who was then the Vice President of Academic Affairs at UMBI, and she gave me Zev's resume, the first word that came to mind was awesome. Yoni read some of the letters. I looked at those this week again, and they're just incredible, and maybe incredibly awesome. So it was right for him to get an appropriate state-mandated raise, except for those of you who know, UMBI was being disaggregated at that time. Usually the promotions happen late in the year, in May, and there was no more budget at UMBI. And some of you know that there were quite a few backyard dogfights over the UMBI budget among the receiving institutions. So, so Zev's raise was not, didn't happen immediately. And he was, as you've heard, when he had something he'd set his mind on and it wasn't happening, he could grow impatient. And so, so I insisted to him this would take a little time, and we had a couple of, probably half a dozen meetings with the system office, negotiating with the medical center, trying to find the money to do this, maybe a few dozen email messages, and things were not happening in the speed that he liked. So he wrote to me and he said, what can I do? Who's responsible for this? Who are the people involved? I said, well, gee, you know, here are the people I'm talking to, and I mentioned the Chancellor, and the Vice Chancellor, Joe Vavona, Irv Goldstein, and the Chief Operating Officer for the system, Joe Vavona. And so, so the next day, first thing in the morning in email, there's an email message from Zev to Joe Vavona that says, Dear Joseph Vavona, and it explains, you know, just been promoted by UMBI, it's our custom to get a state-mandated raise, and I would like to know when the state is going to give me my raise. Sincerely, Zev Pancer. I thought, gee whiz. So, you know, I called Zev immediately. I said, look, let us negotiate this. Let's, you know, this will happen. And he said, well, you know, this needs to happen. I said, okay, let's let it happen. So the next morning at 8 o'clock, I look at email again, and there's a message from Zev to Joe Vavona that says, Dear Joseph Vavona, I request that you respond to my original email. So, it might be to no one's here surprise that this continued for 2 weeks. Every single day, 8 o'clock, Zev sent an email to the Chief Operating Officer of the system. This is a very powerful, important person about his raise. Now, eventually resolved, but I'll say this. The fact that he sent it probably surprises nobody in this room. The fact that if you want to get a raise from Joe Vavona, that's probably the worst approach ever. So, but anyway, it was an interesting sort of anecdote. But then I had the pleasure to be able to interact with him on a scientific level. So after the reorganization, I decided to take a scientific retreat by going back to the lab. And one of the things we was interested in plants, and one of the interesting problems we worked on were these innate immune system receptors in plants, these NOD receptors that Roy and Max referred to. And when I started to read these, I realized that these leucine-rich repeats on these plant receptors were very analogous to these lamprey antibodies that Zev had worked on throughout his career. So I said, well, let me reach out and see if he can give me some advice or something. So it was as gregarious as anyone I've ever met. He invited me up here, we talked a lot, we started talking scientifically, and characteristic, the kind of interaction that Roy indicated, we'd talk in his office, we'd have a cup of coffee, we would argue, and sometimes arguments would get heated, and then he'd say, let's take a break, I need a cigarette. And we'd go outside, and the discussion would be about family, politics, friends, what's going on. We come back to his office and start the argument right where we left it. But what was remarkable about my interaction with him was, I know, like I said, I think that he could be very intense, and people remarked he was a very demanding, exacting scientist. But I found him to be generous. He gave me lots of clones. He gave me strains, plasmids, advice, papers, protocols. And when we finally got the molecules we were interested in cloned into yeast, and I asked him if we could come up to Baltimore to learn how to make these measurements, he said, just send it to me, I'll measure it for you, and if it works, then I can teach you how to do it. But if it doesn't work, I don't want you to waste your time. And sure enough, it did work. We wrote a proposal in the fall. Like a lot of first proposals, it needs to be revised a little bit. But I was looking forward to a very productive and engaging collaboration with him. So like Yoni, I was wondering about some words that might describe Zev as a scientist. And I'd like to read something from the PK Avat that I think is very appropriate. day is short, the labor vast, the toiler is idle, the reward great, and the master of the house is insistent. It is not incumbent upon you to complete the work, but neither are you at liberty to desist from it. I think that really captured, Zev was the consummate scientist, as Hassan said. He lived for science. He was here every morning. He went home at night and he started to write about science, and he did the same thing every day. He loved to engage. So like all of you, I was shocked to learn about his death, and probably like a lot of you, experienced lots of mixed emotions. And I've reached out to a lot of people, especially some people here, and I'm very pleased to be here today and see such a marvelous turnout, especially from his teachers and collaborators. I think that one of the things I've appreciated the last couple of weeks is that I probably didn't know him as well as I thought. Yeah. And but the other thing I know for sure is that I'm going to miss him, and I really miss him. And so my prayer for today is I hope he found some peace. And I hope all of us can find some peace as well. So thanks very much.
Thank you.
So I said at the beginning, and this has come up during the seminars as well, we are really fortunate and delighted that Dr. Xie Hong and Professor Li Mao will be continuing the work. You heard about the transition of Zev's work from fundamental research towards biotechnological applications, and that emphasis to look for the biotechnological applications of Zev's fundamental findings will continue under the guidance of Professor Mao. So thank you very much. Professor Mao.
If I can say a few words, I started to interact with Zev about 2 or 3 years ago, and I was introduced by Kurt Seven in school of medicine, and I heard the shocking news when I was overseas. I'm really shocked. Initially, I tried to find out what's going on. I come back. Every day I'm thinking about. Because before I left, actually he called me saying we need to talk something. I didn't get a chance to talk to him. I really feel something. And as everybody knows him, he's very intense, but he's truly devoted. Science really, he wants this discovery translate to something for the benefit of human beings. So with that, I can here promise you this: I'm going to carefully evaluate what I have left from him. I'm going to do my best to carry this on to see if we can make this wonderful discovery to some benefit of human health, and probably will call some collaborators and previous medical Dr. Cooper and Roy. Probably will interact with you down the road to see whether we can continue this collaboration, because I think the stuff you're doing also elegant, and I can see the potential. Even we can think about different approaches to be able to identify the molecule which can not only recognize these altered proteins as biomarker, and also for potential therapeutic purposes. So thank you very much for everybody coming for this scientific presentation to remember Zev. I really
thank you for the work you are doing, for the work that you're going to do, and for keeping the science alive.
Absolutely.
And being Zev's contribution to our science.
I will do my best. I promise you.
I can tell by the determination you have. Determination reminds me of the red of Zev.
Okay. Thank you very much.
We have a light lunch waiting for us outside, and that will give us an opportunity to keep talking. I think I would like to conclude this research symposium celebrating Zev's life and his research contributions by asking you all first of all, thank you for being here. And secondly, please join me in thanking our two outstanding speakers again today.